on that. You know, the biggest thing for them is I know, I mean, just from having talked to her before about this kind of stuff, is that she wants to make sure there's follow through on this. That you know, we've got a plan to take what we did and translate it into an actual proposal.
Right.
So I would say, like, to me, that means that the overall objective is whatever the research objective is we just sat on.
Right. So yeah, got it. I guess just making them as concise as possible.
Yeah. So for milestones, do you guys want to? There's a couple of things we could put on there. So if we're going to apply for that JGI thing,
yeah,
that can be a milestone.
Okay.
So we can say, you know, apply to JGI CYSP new investigator program.
Oh, okay.
And then, you know, identify funding target.
I think we
have
have to put
listed, but like that's part of the process, like identifying which one we're actually going to apply to. And then after that, you know, we could just have a draft proposal, meet as group.
January, Santa Clara.
Yeah.
Yeah. Submit final proposal.
That's what I'm hoping for, just because I don't want to have to.
Yeah.
So, you know, this is, I don't know, it depends, it's sort of dependent upon what this one is, right? What the funding target is.
Yeah, don't we need to identify it in this abstract? I thought that was one of the, that was the point.
Is that one of the things?
That was point 3 in this abstract, was identify external funding grant target and its proposal deadline.
I see.
So that's something we need to do now.
Yep.
That's why I listed those.
We don't know what the deadlines are for all these, right?
Anything USDA is going to be completely up in the air, and I think Susan knows that, and it's probably going to be fine. And I mean, USDA is the proper place to put something like this.
Yeah.
So I think the NSF NIFA is probably our, either that one or the agricultural microbiome one.
Yeah, either one of those. You mean signals in the soil?
No, I don't think we're going to be eligible for signals in the soil.
Okay.
That one was the remote sensing.
Got it.
Oh, okay.
You should just delete that then. I'll delete that.
So is there another NSF NIFA joint one that we would go for, or is it going to be agricultural microbiomes?
I thought there was another.
There was another one, I think, from the presentation. I think it's
Yeah, there's just it's just the innovations at the nexus of food and energy, and water.
Okay.
That would be the other one. I'm actually not so sure if what we're talking about is an innovation.
I'm also not so sure about that, because we might be getting rid of the water element. Like,
yeah, but well, I mean, I don't know if we need to be at the nexus of all three of them. If we were at the nexus of two of them, but then again, I'm not sure if we're at the nexus of food and energy.
yeah, right. There's no energy element.
This doesn't have to be like totally final though, right? Like we're just putting some potential grants that we're thinking about for the September 30 deadline. We could probably just list some of them and then kind of hash this out in more detail later.
I think we could just choose the agricultural microbiome one for now. I think that's a good one.
Yeah.
And then we can.
Yeah, I think so too.
Sounds good.
All right. All right, good.
Yep.
So now we can remove that. Is that the right date? The 26th?
For T Superb?
For JGI.
Isn't this happening before the submission to C Superb? Is
Yeah.
that for this?
Right.
Yeah, that
TGI
sounds
is
right.
the 26th.
Yeah.
Yeah, so by the way, I got stood up today by the grower, but we're going to meet up tomorrow. So I'm going to get samples tomorrow.
Oh, good.
Like I wrote in the email we discussed last time, we're not sure that soil samples, yes, fruit samples, we vacillate.
Yeah, I mean, I talked to, as I posted, I think on the, on Slack today, I spent some time, I met up with some friends for drinks on Friday. One of them she works for the USDA, does a lot of soil microbiome work. And we were chatting about the JGI metabolomic stuff. And I think that Bill would probably also say this, but they, it's still pretty experimental, like what they're doing. Like they don't have a strong sense of everything, but I actually think it's a really good place for us to be because it is a very progressive, innovative thing to be trying to make these connections, and it directly relates to what we're interested in.
Mhm.
But for that JGI proposal, my guess is that like if we just talk about trying to associate community with metabolic outcomes, like that would be enough for them. And if we threw in like one plant tissue, that would probably be good, but we don't need to. I'm not sure like how much they're expecting us to have like 10 different plant tissues or whatever, you know. But we could easily try to include multiples and see what happens.
The other thing that I mentioned when Matt was around was related to doing an elemental analysis of the soil too. My question, maybe Nate, if I know there's an elemental metabolome, I guess there's some people that does both of those things together. I was wondering if the GDI would include elemental analysis in addition to metabolites.
I have to.
That would be important. It's important for the context of nutrient quality in plants. Eventually, we have to look at the soil elemental content for that. So the question is, do they do that? Is the first question, and if not, then I can reach out to my former colleague that does some of that.
It wasn't obvious from the JGI calls that was one of the components of the new investigator grants.
Okay.
Sorry, I didn't mean to step on you. Go ahead. Please continue.
No stress. I also have, I spoke to one of my colleagues in Earth and Environmental Sciences here who also said that we have the equipment here to do the soil analysis if you want to talk about mineral content and so forth. So it looks like we've got abundant resources for that, but I'm not sure JGI is the right place.
I was thinking about soil tests, just general soil tests too, which is what I think you guys are talking about. Am I right?
Mm-hmm.
Yeah. Elemental analysis is just a soil test, right?
Yeah.
Yeah. So I was thinking about that and didn't mention it last Friday, but I think we should definitely do that.
Yeah. So something that not related then to JGI and our immediate needs, but definitely something to stick into the long-term full proposal.
Absolutely. Although I think, John, you were saying that you could even get a few samples thrown in now. So if we were to get funded by...
Right, yeah. So I just talked to one of my colleagues and he said he's willing to run some samples at no cost. I don't know how many, but we at least run a few samples to get some elemental data from the soil samples that we have as part of the pilot, as part of some preliminary data.
Do you know how they need to be? Can I just like freeze them?
Yeah, if they're not, you could just freeze them because they won't. You can't destroy or create elements. So just put them in a container that won't get contaminated.
So as long as they're not biologically active, that's all, right? Because they will still have bacteria in them, right? So bacteria can affect that.
Yeah, you could just freeze them, and I don't know if that's even needed.
Cool. But not put them in like some of them. Like I have to like the rhizos to get the stuff off the rhizosphere. sphere, thank you, to get the rhizosphere sample, I'm supposed to, from what I understand, I'm supposed to put them basically in like a falcon tube and like shake off all the soil so that it settles out.
Okay.
So in that case, you know, that wouldn't be good for an elemental signal, right? Because that would like, you'd have a fraction that would be in the water and a fraction that would be in and possibly bound to the soil.
Is what they're gonna. So I think my understanding is they're gonna take a sample of it and basically acid digest it. So I think there's this, as long as we can get a representative sample.
Okay, so would you want me to do the same thing for that rhizosphere sample then? Like, would you want me to? So like the soil, I just put it in a container, right? But if I'm gonna do the rhizosphere one, basically I gotta like, you know, take the root and like rinse off all the dirt into the thing, and you have to put it in PBS to do it. I guess I could just put one, not do the PBS thing and just put it in there and freeze it. But then, yeah, okay, I'll figure something out. I'm just trying to make sure I grab whatever is useful while I'm out there.
Yeah, I can also double check if there's a specific process that's required. But my assumption is that as long as you're not contaminating it with additional elements, which you will, I guess, if you use PBS. As long as it's a known quantity, I guess. Once we can you know, calculate it out or discard it from the information. If you have a sample of the PBS as well as a kind of a background.
All right. So should we focus on JGI first, since it's a sooner deadline?
Sure.
Yeah.
We've got an abstract going, which is useful. If you take a look at the agenda for today, we were just talking about immediate research, the very top item, sampling. And does anybody know, so related to the JGI proposal then, but also to immediate sampling needs, has anybody figured out for metabolomics how soil samples get stored before shipping, presumably, if we get the JGI funding? Do those need to be frozen, for example, or is it just dirt in a tube until they're ready for it?
Usually, well, yeah, for the metabolomics one, huh? That's a good question.
Yeah, because I know what to do for the metagenomes. We buy the kit, we extract the DNA, and we ship them the clean DNA. I didn't look very deeply, but I didn't see any immediate advice from JGI about metabolomic samples. They had all sorts of metagenomes, transcriptomes, processing, quality control.
Yeah, because they're used to doing it. The metabolomic stuff is still pretty new, I think, and that's probably one of the reasons why.
Okay.
Let's see. Yeah, I mean, when there's sample prep guidelines, they don't have anything for.
Right.
Are they expecting folks to just send them soil and they do the extractions then?
That'd be my guess.
I will follow up with them about that.
Yeah, you know, why don't you just reply to that one thread that we have going?
Yeah.
With the JGI person.
Yeah, Susanna.
Yeah.
Yeah.
She'll probably.
Small world. I met her at a conference about 8 years ago, and when I was new here 7 years ago, I invited her to give a seminar. So I actually know Susanna.
Nice.
But anyway, yeah. The next questions about the JGI proposal then, as you can see, are details what they're requesting. The bottom of the first page of the agenda, how many samples, and then it would be great if we could all pick, as appropriate, a section or sections of the proposal to draft for the rest of us. And this is why I'm proposing having this Wednesday meeting as well, is if in the next couple of days we can draft the description, justification, utilization, community interest, relationship to the DOE mission, sample preparation, 6 different sections. 1,000 characters each, so not long.
Yeah. Yeah, they're usually pretty. Oh, there it is. Um,
So yeah, bottom of the first page to the top of the second page of the agenda.
yeah.
So I guess fundamentally we had talked about doing metabolomics, right? So we don't have to worry about metagenomes and transcriptomes because we can only do one.
I think you can do more than one. So you can split your time, like your space. Like they're going to give you a certain amount of effort, and we have to we need to ask. This is a question we need to ask Susanna: is like how much can we split it? Because like you probably want to do the metagenome alongside the metabolomics because that's the thing that's. I mean, ideally you might want to do all three, but let's just pick the metagenome in this case. Because I think the metatranscriptomic stuff is going to be harder for us anyway. But I think what we'd want to do is try to figure out if we could do both of like the metagenome and the metabolomics.
Yeah.
Okay, together.
Susanna's email read, "That's fine. You can combine capabilities as long as you're within the limits for each." What that means, I don't know, but it makes it sound like you can propose both.
Yeah, so I guess if you're within the limits of both, that'd be 92 metagenomes.
Yep.
And 50 either 50 polar metabolites or 150 non-polar metabolites.
And polar probably makes more sense because that's the stuff that's actually going to be water soluble.
Okay.
Yeah.
So then, yeah, so we could do, say, 50 metagenomes paired with their related 50 soil samples for metabolomics.
Yeah.
We could get more metagenomes, but I don't know why.
Well, will we have 50 samples? We'll have 50 soil samples. What is are we just getting soil samples from Monterey Bay, or are we going at multiple locations?
I think it'd be good if we could get like a tomato field somewhere else. We don't even have to stick with tomatoes. We could do whatever, but it'd be nice if we could have a comparison that was geographic because, instead of doing a case control, we could do a geographic control.
Absolutely. Greg, are you growing tomatoes? You got them? Okay, so Greg, you're muted.
And
I said I have them, and they're in raised beds.
Okay, you have them in raised beds.
Yeah.
So perhaps Greg and I could do some parallel sampling. Maybe we'd have to talk about exactly how you do your sampling, and so we could do it in the same way.
I'm gonna find out tomorrow when I get to the field.
Okay.
I'll tell you after I try.
That's the benefit of going first. And I can get soil as well if needed.
From a tomato field?
No, but from Fresno.
Oh, okay. Well, yeah.
I don't think we currently have tomatoes, unfortunately.
It might be like, if you do you guys have extension officers around? Like maybe you can ask somebody. I can also, what I can do is I can ask the leafy green, because this guy's a leafy green. Let me talk to the grower tomorrow too and see if he has any suggestions. Because maybe he'll be able to be like, you should talk to this association and they'll put you in touch with somebody. Because, there's a lot of grower associations where they might be willing to let us come in, especially at the end of the season, when. Now, we should also realize, right, that there's a developmental component to this, and we're picking it up, like, at the end.
Oh, yeah, exactly. But, I mean, there's only 3 days, so we kind of have to it seems to me that you've got something going, and we could potentially go out to Greg's plot and just pull some soil. You pull 25 samples, we pull 25 samples, we freeze them, and...
Yeah, I was only going to pull maybe about, like, 10, probably.
Okay.
Like that. I looked at this paper. I've been trying to figure out like what the right spatial scale is for an ag farm, right? So much of microbiome is determined by the soil types and how variable they are across space.
Okay.
And what it looks like because agricultural fields are relatively consistent, usually you're looking at like data which is autocorrelated across like 60 to 100 meters. So, depending upon the field, you could see some gradients across the field, but you know, it's going to not be wildly different. It's going to be more the variability in soil types that's going to be driving, it looks like, what we would detect.
Okay.
The farm manager I spoke with here at Fresno suggested that there will be position effects, for example, based on watering. Edge of the field might be different than middle of the field. So that might be something to keep in mind when you're sampling.
Yeah, and like that was me like kind of estimating kind of I mean they're based on individual studies done for 1 year, so you know, but it looks like you have across your average field about, like you said, there's going to be variability across the field, but it's probably about like 60 to 100 meters. So I was going to try to spread them out, you know, maybe, I don't know how big this field is, but I'm going to try to go like, you know, sample every like 10 meters or something. Or 20, 25 meters or so. That's what I was thinking. So.
Well, we're going to have a whole lot less. Greg, I assume you just have like 1 row or something like that.
Yeah, it totally did.
So,
but it could still be useful.
oh yeah, absolutely. Yeah. I mean, for this preliminary stuff.
And the other question I had was, we're just going to get it around the tomato plant. We don't want like stuff that's away from a tomato plant that's kind of uncultivated, because I'm remembering some people saying uncultivated soil as well.
I think that's a useful control.
Could we do that? Because we have plenty of that.
So I'm just trying to think about the sample. Like we can do 50 samples. Is that what it sounds like? So if we do like a dozen control, a dozen, or I don't know, maybe that's not the appropriate number, but 25-ish at each site that contains both control and the uncultivated and cultivated. Does that sound like what we're moving toward?
Yeah, I was thinking, so the thing I was going to suggest was to do something that was sort of proximate to the plant, either directly below it or directly adjacent to it, that wasn't in the root structure.
Yeah.
And then a sample that was in the root structure, that was the rhizosphere. And then one other thing, and that could actually be the roots. That could be a fruit. That could be a leaf, a stem, whatever. A root would be easy, but that's another option for us to think about, like how we want to break them up and what samples, and if you wanted to add in another sample that was even more distant from there. So if we start adding more samples, right, then I would suggest we cut down on the number of plants we sample and only do like 5 or something.
Yeah, I think that makes sense. Yeah, you do five plants, 1 outside sort of the root system zone, 1 in the root system zone, and then 1 completely outside the tomato field, if you will. And then I don't know if we want to do leaves and things like that, we could, but
I guess I would vote for not plant tissues at this point. Unless we know that we've got the same cultivar, which is the conversation that keeps cropping up. Yeah,
we won't. I'm sure that's not going to be the same cultivar.
Yeah.
I don't even know what we're growing. I know they're tomatoes. I didn't plant them, so they've just been there.
And Nate, I don't think we should do the rhizosphere as in, oh, now we're going to like tear up the plant and grab the roots and try to knock off the soil. I think that's just going to be too much work for right now. Let's just do more or less bulk soil, outside the root zone, and bulk soil within the root zone is probably going to be good enough. We've only got a few days, really.
He told me I can dig up the plants if I want, because he's done. So.
Oh, okay. I don't think, Greg, I assume you're not done.
We can replant.
That's the spirit.
Yeah, I mean, I was just going to buy some new plants. So if we want to rip out a plant or 2, I'm good.
Okay.
Yeah, I mean, the cool thing is that you'll definitely see a difference, right? So we know that the rhizosphere community, so if we're just looking for differences and we're trying to identify, like, are certain communities associated with different types of metabolites, I do kind of think it's not a bad idea to pick some communities that we know are going to be different, so that if we're looking for differences in metabolites, we might actually observe them, because we would expect the communities to be different. Does that make sense? So that was one of the reasons why I thought, oh, this might be good, because then we can compare like something we know that's and the rhizosphere has been studied a lot. So
Yeah.
Well, you're going out there tomorrow, right, Nate?
Yeah, and I mean, listen, we'll see how it goes. And maybe
Exactly. Yeah. Sample whatever you sample, and then you tell us what to do. Do the same
Yeah,
thing.
that sounds right.
I'm just gonna take everything I can.
Okay.
Just go from there.
So
And all this soil goes into like a -20. Is that what you're doing?
I would put it in a -80.
-80. Okay.
Just to be safe.
Okay.
and just to be and this is the metabolomics, metagenomes, and the microbiome, potentially elemental analysis, right? So it's gonna be four different things.
Yeah, so this would be well, the metagenomics and the microbiome stuff is this, I guess, the same stuff, right? Like the genomic component, the DNA component, the polar metabolites, which is what we're thinking of, right? And then the soil analysis. Is that the three things that we're sort of targeting?
Sounds good.
Okay. So I'm just going to put them in Falcon tubes and
Yep.
do it that way.
And freeze them.
Yeah.
So just a note, the we can get DNA extraction kits easily at Fresno. So I could do potentially the soil extractions for DNA if we get the JGI grant and they need us to ship it to them. But James Schmidt said that we can't do any shipping costs on our $1,000 each. So any shipping we're going to do is going to be out of pocket or some other source of funding.
I can ship you my sample.
Yeah, it's fine. I can ship.
Yeah, that shipping's no problem.
Just wanted to point it out.
I mean, how much is it?
Not much.
Well, it's going to have to. You're going to have dry ice in there, and so it's not. You can't just put it in the.
Yeah.
Yeah. It's super expensive.
Yeah. Overnighting coolers with small coolers, styrofoam containers with dry ice and tubes.
You can just drive in your car and get a transportation reimbursement.
For that.
It could be like a meeting.
Fresno.
Right? A meeting.
Yeah.
I'd be happy to drive over to Monterey Bay.
I was just there.
No one stopped by to say hi.
I thought of you as I drove through it and stopped at the REI.
Right. So we're going to have Nate and Greg in charge of identifying, once sampling's done, that first step. How many metagenomes and metabolomes are we requesting? Because that'll be dictated by our sampling.
Yep. So Nate, you get a maximum of 25 samples if we're going to try to match you.
Yeah.
Yeah, we don't have a really big
I'm going to
feeder.
try not to go that high. I mean, I think if we're 5, we're all right. Like,
Yeah.
Okay.
I mean, I looked at that PNAS paper and they only had 4 per site. So whatever,
Yeah. Well, okay.
And then what about another big question in my mind for sampling has been volumes. I know we only need 25 milligrams for soil genomic extraction. I have no idea about metabolomics or soil nutrient mineral content. So hopefully somebody else has a good idea about that.
I mean, I'm thinking if you get a 15 mil Falcon or a 50 mil Falcon full of soil, that's going to be more soil than you're ever going to need in a lifetime.
I was hoping.
Yeah.
That's more than enough.
Okay. Okay. So how would we like to split up the writing? Do people have time in the next few days to grab one or more sections and either individually or jointly draft?
I can probably do one tonight.
I would be good. I think the ones that we're going to have particular issue with are going to be utilization, community interest, and mission. If anybody particularly feels like they would be good at one of those 3.
I could give a stab to community interest.
I was hoping you were going to say that.
You want me to? Utilization is just how we're going to analyze the data
I'm not.
and what it's going to be used for. Yeah, I can do that one. And it'll be used for the grant proposal, and I'll talk about data availability, all that sort of stuff.
Well, we're using it for like what do you call it?
Preliminary data.
Preliminary data, yeah.
Yeah, but I'll also say, like we're going to put it on JGI's database.
Maybe there's a paper.
Publication. Regional differences.
Okay, I can draft sample preparation as much as I know about it.
Yeah, the soil samples, the only soil samples I ever sent out for that kind of metabolomic stuff, I've always just sent the soil.
Mm-hmm.
I've never prepped it.
Yep.
So I don't know what they do.
So maybe it might make sense to have basically from each site to get 2 soil samples, or at least split it in half, where half the soil gets mailed to Joe and the other half just sits in our -80s and in the hope that we, can then send it to JGI.
Mm-hmm.
We just have to match up each sample so we know who went with who.
Yeah, well, we actually need 3, right? Because we need to send a sample to John too, right?
Oh, right. Yeah. Right.
Hence my comment about shipping costs.
Yeah, but the stuff we got to send to you, Joe, like I can send you a microcentrifuge tube of stuff. Like,
Yeah, exactly. It's fine.
like hardly anything we have to send you for that kit.
Yeah.
It's like a half. It's like a half a gram or something that you need, I think, for the Kyogen kit.
Tiny amount.
Yeah, I'm sure the elemental stuff is pretty small too, but I'll double check the actual quantity needed.
Yeah, I will say that like there is some stuff, microbiomes where we do want to kind of be as consistent as possible with how we're handling them, like how long we've kept them frozen, what temperature we've had them frozen at. So if you guys prefer me to put them in -20, I could do them at -20 instead to make sure that it's more consistent.
No, -80 is fine. I just, like you said, we just need to be consistent. So if you tell me exactly what you do, then Greg and I can grab them and put them under exactly the same conditions that you have.
Okay.
Let's see. Oh, we don't have
Elementals analysis. When I've done soil tests, you usually do an average across a whole row or a whole area or a planting field. So you take like 3 or 4 different holes and you kind of scrape off the sides so you get like a mixture of the stuff. Is that what you want, John? Is that what you're thinking?
I mean, I guess if we can correlate it with, because I think it has to correlate with the other data.
So you just want it from the same dirt, the exact same dirt.
I think ultimately we want to correlate the elemental composition with these other endpoints. So I think treating them the same way.
I just know that when I do soil tests, I take it below the surface for sure. Like usually about a foot down, something like that.
I don't think we want to go that low for this sort of stuff.
Like 6 inches down, or I mean, I'm just trying to think about mechanically. What are we doing?
Nate, are you going to take like one of those little hand rototillers out there and just pull plugs?
I got a shovel.
Okay.
I'm gonna get underneath that plant. I'm gonna roll it up, and then that's about as deep as I'm gonna go. So I'll tell you about how deep I go once I get in there. I've got a trowel that's got markings on it, so I can tell you approximately how deep I go. But I'm not gonna go super deep, right? I don't think we want to go. We want to be around where the root structure is going to be, right?
Yeah.
I presume. So.
Okay, that sounds perfectly scientific. That's perfect.
And Greg, I'm not doing a traditional soil test. It's just an elemental test, which is not
okay. I don't know anything about that, so I'm just trying to figure out.
So you don't need to do the traditional soil test method. We're just trying to look at elemental.
Okay.
Yeah, I'm looking forward to learning more about all of this at some point.
Yeah, it's like all new to me.
Yeah, I would volunteer to write something, but I don't know about the expertise.
Right.
I'm trying to think if there's anything else where I could contribute. I don't know if there is.
I think description and justification should be almost straight from the abstract that we've written in some sense. Not necessarily specifically for JGI, but that is what you put in an abstract, is the description and why you're doing it. So,
right. Okay.
In theory, anybody could write those, or at least draft them, because then we just need to have community input from the group.
Yeah, we can just pull them from the abstract. I guess, yeah. If that's the case, then I can try to take a stab at them.
Thank you.
So John, you'll do the description?
Sure.
Was that a sure?
Yes. Yeah.
Okay.
Or we can work on it. I mean, yeah. I assume that we'll have one document where we're just putting this all together.
Yep. Just add it lower down on this document.
Got it. Okay.
Joe, I've got to go, but so our due dates on these things are Wednesday morning when we have our next Zoom.
Yeah,
call.
let's do that.
What time is our meeting on Wednesday?
10 a.m. I think.
10.
Yeah,
Okay.
10.
Yes.
Yep.
Yep.
No meeting.
All right, folks. I will work on that, and I'll see you all at that time.
Sounds good.
All right,
Have a good
thanks,
evening.
Matt. So Nate, as soon as you get those samples, if you could send us an email, Matt and I both, what the deal is with how you did it, then we can go down and do it in the food project.
Yeah, I'll just hit you on Slack.
Okay. I hope that I have it. I'll make sure that my notifications on Slack actually connect to my phone. This is like kind of a Luddite, so.
Since Bill's
Pulling you into the 21st century.
I know you are.
Since Bill's not here right now, do you think it's good to nominate him to do the DOE mission statement?
Yeah, I think that's fine.
He's in a teachathon, whatever that is.
I will email
Sounds
him.
exhausting. But rewarding.
All right. Sounds good. Let's see. And it looks like everybody's been switching to CSUPER briefly so we can all enjoy our evenings. Looks like everybody's been making their contribution pages, which is good.
I think I'm missing mine.
I haven't done mine yet.
I need it.
Oh,
I didn't know that was one. I tried to just.
That's okay. But we're hoping by the 28th, so you've still got 4 days to get that put together so I can compile them and send them in.
I got to get a GE into my course consent fast. So that's been my main emphasis right now.
Yep.
All right. So I got to run to, let me know if there's anything else you guys need, and I'll let you know how it goes tomorrow.
Yep.
Okay.
Sounds good.
Thanks.
Luck.
So do we have somebody doing the justification part? I'm just looking at that.
Justification. I think those,
Did someone do that? Did someone elected do that?
not that I heard.
Okay.
Is it gonna? I'm just wondering if that is more about I guess, yeah. Somebody will have to talk about the actual metabolome and the metagenome, right, for the justification.
I would think that would be to the extent, just expanding on what and that's difficult. I'm not saying it's easy, but expanding on what we've written as the justification in the abstract, that no one, as far as we know, no one's ever looked at these connections before in this sort of proposed detail. That's what I would argue is the justification in many more words. So yeah, that's going to be tough.
All right.
I mean, maybe Nate left, right?
Yeah.
Yeah.
But maybe that's also part of the utilization part. Do you think? I'm just guessing, because I've never had to write a justification like that, so I'm not really sure. But the scientific rationale behind that in my head would be that we need this data we're sort of testing the data to see if we can if there is a relationship or if there is some kind of basis for the larger grant, right? Does that sound right?
I'd say that's accurate in my mind.
So I'm wondering about utilization and justification being really close in sort of the kind of information being used to write those sections.
It could be, and it's also like hypothesis generation, right?
Mm-hmm.
Which is a lot of the kind of plans for, I guess, utilization, like what we're planning on using this data for in the larger grant. It's these hypotheses that we think will be applicable in this larger grant, and this whole process is generating data to kind of support that.
Mm-hmm. Let's see if Nate can write that then. That's what I was going to say.
Leaving.
Yeah. And if I get time, I can try to edit the abstract in such a way to move some of that, to shorten it and move some of the excellent language that's currently there down into one of these.
Yeah, I was going past the abstract to the 2-page, what is that thing called? The 2-page.
Right, whatever it was, yeah.
Whatever that thing is.
Yep, white paper or something like that. The thing that C-Superb wants for them to send on to the program officers.
Yeah.
Yep. Yeah. And different abstracts have different lengths. So it's not bad that it's long. It's just bad for sending to C-Superb that it's long.
Right.
But we will definitely save the wording and reuse it as necessary. I think the last thing that I had, and definitely not to cut off discussion, is back to the C-Superb meetup.
Okay.
Just thinking about what we're proposing to C-Superb when we return our paperwork a week from now. Somebody had noted that it's super expensive to get into the Marriott, which is absolutely true. James had told me that when I called him before our last Zoom meeting, that they had also looked into the costs of staying at the Marriott. And for the days that we don't have the group rate, it's whatever they said, $500 a night or $250 a night, or something ridiculous. James said he didn't know what was going on. Usually it's because there's a game, a football game, which is just up the trail from the Marriott, but it's a couple miles. But we're not sure that that's why it's so expensive. I'm going to look into other hotels nearby. There's a ton of hotels. Would everybody be willing to consider meeting at one of the Manita airport hotels if that's less expensive?
What's a Manita?
Oh, sorry, the San Jose airport.
Oh, okay.
Yeah, I mean, I think I'm good with whatever, but I know that both Bill and I are at San Jose State, which is basically there too. So we can always, if meeting is an issue, but I know this hotel itself is the main cost. But
hey, what do you do when you have candidates come into to San Jose State? Don't you guys have deals on hotels right there?
The good question. Yes, probably. I don't know specifically, but yes.
They might have the university might have a negotiated rate at a hotel that we could just all land at. And that might make it less expensive.
Okay. Yeah, I can ask about that. But so to target, so probably downtown San Jose somewhere, which is not too far from the airport.
Yeah. And not too far from Santa Clara for those of us that are going the next day to move hotels.
And it's within 5 miles of
Yeah.
Santa Clara. Yeah.
Yeah.
Where the C-Superb is.
Okay. So if you'll check on that, John, I'll look up pricing on other hotels nearby just to see if anything's way more competitive than the Marriott. But there's no reason to be at the Marriott necessarily. It's convenient not to have to change
You can
hotels.
also at a hostel. Maybe that's another option.
Get a hostel.
Yeah.
Airbnb.
That's funny.
Pass.
Yeah, Airbnb will work.
We could camp. I bet there's a state park nearby. We just camp.
Oh yeah.
There are lots of overpasses that we can sleep underneath.
Yeah, there are.
Yeah, lots of homeless encampments around. Actually.
Yeah.
Yeah, so I'll see if there's any SGSU connections for hotels.
So is that what most of our budgets are going towards?
Like half.
Yeah, so for me, I'm in Oakland, so I probably wouldn't get a hotel. I would just kind of come down for the day.
Right.
So are there other things that we could be applying this budget for that would be helpful?
It's just supplies, right? I think
it's travel and supplies.
Okay.
And I'm pretty sure, depending on how many people wind up deciding to chip in on this, we've got plenty of money for DNA soil extraction kits, soil DNA extraction kits. And we can't charge shipping. So unless we think of any other kits or supplies, we could have a great time traveling somewhere or not waste the money.
Yeah, I mean, I know that my colleague was going to run some samples, but maybe we can like get supplies for that as another way to run more samples, possibly.
Yep.
Yeah.
I'm
all out of pocket.
Definitely willing to consider that. The thought to keep in mind there is that I would need to get them, or someone who's at a campus that has the.
Yes, yeah, you would. Yeah, it would go through Fresno, I would think.
Yeah, and then I'd have to ship them somewhere,
And
which is okay on a limited basis.
Right. I think some sulfuric acid, I think, is what they use. So you can ship some acid.
Yeah.
That might be a little more expensive.
I'm just kidding. But
I'll go over to the mailboxes, et cetera, near my house and see what the situation is on shipping fuming sulfuric acid.
You could send it on an Uber, and then we could charge it as transportation.
Now that's creative thinking.
We actually do, when I was at my old job at UCSF, we had a cohort study where we would be collecting placentas from women after birth, and we analyzed them for chemicals. And they would put it in a cooler with an ice pack and send it in an Uber from one hospital where it was collected over to UCSF campus, where it would be analyzed. So it does work for short distances.
Fair enough. All right, so everyone's got their marching orders.
Think so.
Okay. Sounds good. All right.
Excellent. Thanks all.
Okay, see you Wednesday.
Yeah, absolutely.
Can I just say one thing, guys?
Absolutely.
Wednesday, during the discussion, we will probably have to discuss the issue that was brought up by Nate about the water quality.
Oh,
You
yeah.
think that will be time for that at that time? Is that part of the plan?
I think we can.
I was able to just jump in after class, so I thought I'll listen in to see what's going on.
Yeah.
But if the water aspect is not part of it, I just wonder if there is still a place for me in this, and if not, then I just want to know, because then I can think of other options possibly with some other members of the group. So we need to discuss this as soon as we can, I think.
Yeah.
I was just talking to a friend of mine when I was out of town, and she's I don't know what her title is, but she works for American Rivers Institute or organization or whatever it's called. It's a environmental group that does water quality in the Central Valley. And so she had some really interesting ideas that maybe we don't need to talk about right now, but about how to deal with water quality, how to think about, determining water quality and the quality of the runoff and everything. And she's been working with farmers in the Central Valley, and said that it might be possible to work with some of her farmers are the ones that are like more progressive because they're doing environmental kind of and species restoration stuff. So those are things to think about. Maybe on Wednesday, or maybe Jeroen and I should have a conversation about that.
So on Wednesday, who is going to be there? Is there going to be a higher show up at that time, you think? Joe, or?
Not necessarily.
No?
Yeah.
Okay.
I don't think so. The same number of people then as now indicated that they'd be available, but not everybody showed up today either, which is understandable. It's later in the day. So there's a probability of having more people next time. I think some people some people are less interested in the immediate needs, which are the C Superb submission and the JGI proposal, since the JGI proposal that we've been talking about is a little bit ancillary. It's nice, but it's not essential.
Yeah.
So I suspect when we get back into talking about the project itself, we might get more people back into the.
But do you think that whatever is going to be eventually coming out of it, we could still go along in this group to see Superb? or do we need to make a decision about this issue? Being it's such a short time that we're given, between the IDs lab, and we have this issue since the start that we're with a lot of people, and it was clear that it's not really possible to write a proposal for 12 PIs. So something has to happen. But can we still go to that CSUPERB as a group and then basically use the money to kind of use the momentum and possibly write, some people, I don't know if it's going to happen. I was thinking, I talked with Isolde today, and she also agreed, maybe we can just make a proposal like, we have a couple of ideas about more specified on water to see who in the group might be interested in that part. And that we split up, but that we should still kind of going forward as a whole group to see SUPERB right now. I just don't, I just wonder about that.
I think given the 1 week time frame to give a response to CSUPERB, personally, I think it makes more sense to stick together just for that submission. But I definitely think there's enough people in the group and enough interest in different directions that multiple grant proposals could come out of the group with multiple overlapping sets of authors.
Yeah, and I think like knowing what you guys are, I think you, Joe, also mentioned it at one point, maybe there are some ways that we can link different groups together in some way as a network, but also just knowing what you guys, if it would be that we're going to split this off and you guys are not going to talk about the water as much or doing research in the water part, then still being involved and kind of understanding what you guys are doing is really going to help me and the people involved in it to see how we can make a contribution to the field in that respect. So it would be great, from an intellectual perspective to stay involved in this. And like, I don't know if I don't want to hold on, make things go slower. If it's not workable, then obviously we shouldn't do it. But I'm just wondering, we have a really good capital of brains here together, and kind of.
Personally, I feel like that we can link water back in. I think personally that's a good thing. It's just a matter of how. I think that.
Yeah, it is. There is a very logical connection, obviously, between the use of the land and the water, and what water quality or what water quality means. But I do see also that there needs to be a split point at some point. It becomes too unfocused otherwise. So yeah. Anyway, I'll try to be there on Wednesday to hopefully be able to continue a little bit about that discussion as well, so that I can know which steps to undertake to also get something from this IDs lab and maybe. So.
Sounds good. Yeah, I'll put it on the agenda.
Sounds good then. Thank you.
Awesome. All right.
All right.
See you guys on Wednesday.
See you Wednesday.
Perfect.
Bye.
Bye all.